Okay, guys. So I'm an idiot. I cut the video off that I just made one slide too short. So this video is going to be the following slide after the video that you just watched. So it'll only be one slide, and then I have a third video, a lot of videos for one single lecture. That's only like 9 slides. So I apologize, all the videos, but all the content should be on there after I get this one finished. So I will stop talking about that, and I'm gonna pull up the little whiteboard thing that I did for you guys last time. And I've done the same thing again, kind of giving you the flow chart of our muscle breakdown, starting at the top with what we call the whole muscle, or just muscle, which is broken down into smaller units called fascicles, which we learn are bundles of fibers. Fibers, as we talked, are the fancy way to describe our muscle cells. Inside our muscle cells are myofibrils, which we introduced the last lecture, the last video. What we're going to talk about here in this video are what we call sarcomeres, and inside of, I should say functional units, not function units, functional units of a myofibril. And inside of sarcomeres are the actual contractile things, the things that do the shortening, what we call filaments. So let me stop that and pull up my lecture again. So we just finished talking about the structure of a muscle, fiber, the cell, made up of these myofibrils, and we're going to talk about the filaments and sarcomeres and the parts of a sarcomere here on this page. So let me give you a second here to write this out. Okay, and if you need a little bit longer, you can just pause it. All right, reading through. Within myofibrils are smaller structures called filaments. We see them listed over here on the previous page, thick and thin. They are called thick and thin for the simplicity's sake of one's thicker than the other, one's thinner than the other. They're both about the same length, 1 to 2 microns, so they're pretty small. The thin filament's 8 nanometers in diameter, thick filament's 16 nanometers in diameter. So thick filament's twice as thick as the thin. Both of these thick and thin filaments are the structures that are directly involved in the contractile process. The amount of overlap of these filaments depends on whether the muscle is in a contracted, relaxed, or stretched state. You'll see a picture here in a slide or two that kind of really shows you what that all means. Filaments inside a myofibril do not actually extend the entire length of a muscle fiber. Instead, they're organized into functional units, which we call sarcomeres. Okay, so a sarcomere, as I put there on the whiteboard thing I created, is a basic functional unit of a myofibril. And here in our couple illustrations at the bottom, we show what a sarcomere looks like. I like this illustration because it kind of showing you that we have to remember to think 3-dimensionally. This one's a little bit kind of easier to tell things, especially when it comes to the thick and thin filaments that you can see. But here's what one sarcomere looks like. You can see we're showing one sarcomere from this bracket all the way to the other bracket, which means that it goes from what we call Z-disc to Z-disc. A Z-disc is therefore the border of a sarcomere. These are discs composed of dense protein material that serves as a place of attachment for our thin filaments, which you can see are shown here in yellow or in the lighter, skinnier red lines. Attached to that Z-disc is a thin filament. Okay? Now, our Z-disc happens to be the dead center of what we call the I-band of a sarcomere. In fact, a singular sarcomere is going to have 2 I-bands because you've got 2 Z-discs. An I-band is actually the part of a muscle that you see looking light as far as the striations go. What's nice about the word light, it has the letter I in it. We talked about light, dark, light, dark, light, dark striations. The A band is what causes this, causing a dark striation. So it goes light striation, dark striation, light striation. Now, what we don't show in this illustration is there would be, an entirely other sarcomere here, because there would be a Z disc. So there would be, the same situation over here. There'd be another one over here. The picture on the previous page kind of shows it, but not, the best. We're really kind of just showing one sarcomere in these images. Okay. But this is where the light, dark comes from, light has an I in it, dark has an A in it. So that's kind of an easy way to remember it. The A band is the portion of a sarcomere that goes from one side of the thick filament to the other side of the thick filament. Okay. Now, an A band is strictly found within one sarcomere, whereas an I band actually spans across two sarcomeres. So it's split in half between two sarcomeres by the Z disc. Okay. Now going back to the A band, you can see there's three different zones. Two what we call zones of overlap, which are exactly what it sounds like. Zone of overlap within an A band is the region where the thin and thick filaments overlap one another. Okay. The H zone is the portion of the sarcomere where there's only the thick filament. So you can see the thin filament ends there, the thin filament ends there. So that portion is the H zone. The H zone is also where you find the M line. M line, think M for middle. Okay, the M line is the middle of the sarcomere, and it contains supporting proteins that hold the thick filaments together at the center of the H zone. There is a third filament that we'll talk more about on the next slide, so that will be in the next video. We call this titin. It's a type of protein that gives special properties to our sarcomeres and therefore our muscles. But we'll get into titin a little bit more here in the next video. What I'm actually going to do is pull up myself and kind of talk about the relationship between thick and thin filament. So I was going to use my hands and my arms as a demonstration. So, if I pretend that, my left hand, this is my thick filament, this is my thin filament. We know in a sarcomere that there are portions where the two filaments are overlapping one another, and there's portions where they're not overlapping. But when a sarcomere shortens, what takes place is the filaments slide over one another. And when the muscle relaxes, they slide back to their original position. But when the muscle shortens, the sarcomere shortens because the filaments are sliding over one another. I don't get into it in this class, but in my physiology class, I talk about how essentially the Z discs get pulled closer together because the thick filaments pull on the thin filaments, causing them to slide towards the M line, which is why the theory that describes muscle contraction, it's called the sliding filament theory. So it's kind of cool. Really complicated. A lot of ins and outs go into it, but I'm not going to really get into that for this class. What I do really want you to know, and I'm telling this straight up, this whole picture, which includes this part and the parts that are labeled up there, which are the exact same things as labeled down here, just kind of looks different. Every part of this image will be fair game, and you will see this image, really the two images together. You'll see it on the exam. So make sure you can identify the discs, the bands, the zones, the filaments, the M line, that kind of stuff. So put it to memory. Okay, I promise this should be the video that then we'll pick up in our last video with myofibrils and the proteins that make them up. So I will stop here, and you'll watch the rest in the third video. Thanks, guys.